Thank you for the invitation for me to share the recent study we performed to evaluate can we do the T2T human genome assembly using ONT-only platforms. And this work is done by the team at the GIS, consists of Melis, Prasad, Yushipa, and Dewey, and through the collaboration with ONT. Allow me to give a very brief introduction. What do you mean assembling? Assembling is a computational procedure to reconstruct the genome sequencing using fragmented DNA sequence data, or called reference, called the reads. Use a human genome as an example, right? We first generate the reads and overlapping and cover the whole human genome. And the color of the two, the reads representing their so-called chromosomal origin or parental origin of the reads, right? There are two types of assemblies. In the early days, people trying to sample the one single consensus sequence of a genome. We call the primary assembly. More recently, with advanced technology, we'll be able to even further to phase this assembly into the hepatide assembly, representing the two copies of the each of the chromosomes. And well, there's a lot of applications about de novo assemblies. We are currently using this to generate the human reference genomes or pan-genome reference. And also for many other species, there's never been done sequencing. This de novo assembly is usually the first step to get into the understanding how about the genome sequencing. There's many tools. They can do the de novo assemblies, depending on what kind of data, the kind of your sample size, and what kind of a computational resource do you have. And this is basically a snapshot, say, what has been done for the human genome assembling, right? The first milestone is probably released the draft of human genome sequence back to 2003. It is the first milestone. After that, then I will see another milestone is probably happen almost 10 years, 20 years later. We'll be able to do so-called the T2D assembly of human genome, where we can see that we now have a complete chromosome structure, but still have a certain region of the human genome has not been fully resolved. That's why there's further improvement. Then with the technology progress, people no longer is both beyond the single genome assembly, they go to the pan-genome reference, trying to capture, recharacterize the genetic diversity of human populations using the T2D kind of strategies. And to do this, usually they will require a certain amount of data, high quality long read data. Short read would not be able to do this. We understand that. You probably also need a very called ultra long reads to be able to allow resolving, some of the, contigs, right, to connect them into the bigger contig. They sometimes using chromosomal chromatin information to be able to do long range phasing. Not only talk about the contig, or if you have parental information, you will use that one. There's a common different algorithm. The most common one currently are being commonly used is one called Hyphesium, right? And then one is used Vercal. They are based on slight different methodology for building the so-called assembling graph. And so this is basically the short snapshot of a major step when you do assembling. We look at all the de novo assembling algorithm, they always have a certain step to correcting the reads, to clean up the reads before they build in this graph. The graph is based on what? Based on the so-called overlapping information of different reads. They connect reads together and figure out this called the pairwise connections between all the reads. And this can be built on the two principles, either using overlapping sequencing between two reads, or they look at shared k-mers, be able to build the connection between these reads. Then you build this giant graph. Then you have a mathematical way to say, how do I find the path going through this graph to capture all the pairwise information, then give you this consensus genome sequence supposed to reflect all these reads, how they overlap each other. Then the ultra long reads are very useful in the way to finding this path along the graph. And this is have a short summary. What kind of data has been used, to generate the human, de novo samplings, from even the first one, T2D CH13 has commonly referred now. You can see from this. This is definitely not all the studies. There's a lot of studies that has been done. This is only a snapshot of a few examples. But you can see from this table, most of the almost all the effort are currently using called a hybrid approach. Basically using different technology platform, generate a different kind of data, allow this T2D assemblings. The question now, given all these things right now, will be possible to do the T2D assembly using singular platform. In this case, we ask whether can we do this using oriented platform. So this is a pilot study we performed recently. Yeah, we're choosing a sample from our Asian reference genome project as a trail, as Indian trail. And we did assembly for the child. We use parental information for phasing, but the de novo sampling was truly only performed using child information. And we generated data of a duplex, ultra-long, and POSI. This is a summary of how much data, but we're not using, the full data sets. We're using a medium range coverage, talk about 40x duplex plus, 20x ultra-long and 30x POSI to do the initial evaluation of T2D assembling. And we explored two assemblers. One is vertical, one is high-phasium. Here is basically the data quality information. Gordon just showed you, what is a simplex, duplex read quality. This is the data generated at the GIS. This is read from our lab. You can see that the duplex is quality in terms of quality is very similar to hi-fi now, but definitely still a little bit lower. This is basically, you can look at the curve, and this is a red, a duplex. The blue is actually the hi-fi, okay? And they're similar, but it's definitely not the same yet. It's definitely similar. So this actually gave us hope. Say that maybe, purely ONT-based de novo sampling can work because the accuracy is really catching up with hi-fi. So this is actually just be a very brief presentation of the outcome of this de novo sampling using 40x duplex, 20x ultra-long, and 30x POSI. I guess this is a favorite chart usually commonly used by the ONT communities, but it's very hard to interpret it unless you're very familiar with chart. I would encourage you to look at the table in the middle. That'll be more strict. Basically tell you how many scaffold or contigs you can generate using two algorithms, vertical and high-phasium. And we present result for HAP1, HAP2, basically represent two pools of the contigs. They are called phased contigs, right? And you can see the size roughly close to the human size. Give you a reference for CSM13 is basically talk about 2.96 for HAP1 and 3.05 for the HAP2 in terms of length of a human genome. So in terms of overall length, it's very similar. It's catching, similar to the, CSM13. And if you look at the overall outcome, you can look at the N50 basically mean, the medium, middle size of in terms of length of the contig, right? It's about 134 or 100 over 100 of megabase, okay? And what I like to see is that this is a table to give you more information about outcome. Basically, on the left side, your left side is an outcome from vertical, and on the right side, you can see on the high-phasium. Okay, if you compare these two algorithms, well, the high-phasium will generate a little bit longer genome assemblies, genome assemblies. But if you look at the length of the contig, then vertical is probably a little bit longer than high-phasium. Okay, and if you look at the quality, high-phasium seems to do a little bit better, both in terms of switch errors because we do a sampling. Sometimes you missample the different chromosomes can tick together, and this is called phasing arrows. And you also look at the overall base quality, how actually the sequence that you assembled out. Then high-phasing seems for both criteria is a little bit better than vertical. But if you look at the timing they need for these two assemblers, this is very different. They start with the same data set, like I said, 40x duplex, 20x ultra long, and 30x pulsing. This is the time. Use a vertical, you're going to take you 200 hours to get job done. But if you use a high-phasium, 12 hours, you'll be able to finish the whole analysis. You can see the speed between these two algorithms is dramatically different. This is have a significant consideration when you're trying to do pan-genome reference, analysis. This is really matter. This is a presentation of the so-called visual presentation of contig or scaffold you be generated from these two algorithms. On the left side is a vertical, on the right side it's high-phasium. And you can see that for the vertical, they try to sample the whole chromosome out as much as possible. We call this scaffold steps. They have a scaffold a step inside, right? And of course, you can see that you realize there are 4 chromosomes are tangled together. They call the chromosome 13, 14, 15, and 21. The reason is because this is called rDNA cluster, because this is a sequencing that are highly repetitive, multiple copies. Probably it's called 100 copies, and they're concentrated in a certain part of the genomes. Usually, cross-centric chromosomes on tip, they're concentrated in the cluster there. These sequences are very hard to be resolved. Okay? Even at this data level, we will not be able to resolve in this cluster sequencing. That's why these chromosomes all put together. We are trying to sample them. They put them together because they cannot configure out which sequence comes from which chromosome. They all seem together. Because for the high-phasium, because they don't want to do this chromosome level scaffolding, so you don't see this cluster because they leave. If I cannot figure out, I leave them alone. Okay, you have a much more shorter contig here. You can see label them out. For most of high-phasium, you can see that very rarely you can see one chromosome has only one scaffold or contig. There are more than one, because they don't want to truly sample them out. And so that's why, but they don't have tangle. But for vertical, they're trying to do this. Of course, even for vertical, they still have called the tails, a small contig. They will not be able to incorporate into the sampling. This is called the difficult region. I would bet most of this smaller fragment or small contig come from these difficult regions of a human genome, like centromeres, DNA clusters, or other highly repetitive sequencing. That's why they cannot easily be resolved. Mitochondrial, yeah, you can see both of them are trying to sample the mitochondrial genome out. You have to remember, this is very artificial. Why? We understand, with the human, cells, there's hundreds of thousands of copies of mitochondrial genome. There's a lot of, we call the somatic mutation there. But whatever they translate for vertical, they just give you one consensus sequence. For the high-phasium, they still try to phase them. So they give you 2 consensus contig of mitochondrial genomes. There's another way to look at what's called the T2D view of this chromosome, right? And on this plot, the color represents the parental or maternal and the paternal chromosome. They be resolved, being their result. If there is a white color, basically means that piece of sequence has not been resolved through this exon. We call the gaps, okay? And you can see all the centromere is still remain unresolved. Yeah, true. And then for many of the acrocentric chromosome tips, you can see where the rDNA is cluster located. They are still largely has not been fully resolved either. Okay, and we do see for many chromosomes, we reach to call the T2T. Basically, we see the telomere sequence on both ends of the chromosome because the black dot is presenting we do see sufficient number of the telomere motif presenting that the sample really can reach to the end of the chromosome. But not all, but for many of the chromosomes, we'll be able to achieve T to T. This talk about phasing accuracy. Huh? When you talk about, without so-called phasing exercise, you just do direct de novo sampling with the duplex plus ultra long, you can end up with so-called contigs, right? The middle of a reads come from both parents' chromosomes. You look at, because this is called the camera plot. You look at the contig, right? Is your camera come from paternal or maternal? Huh? And you can see that most of these contigs is a mixture of the reads come from two parents. And if you put a POC, long range interaction information, configuration information, then you'll be able to clean up this called assembly. Then they have a proper phasing of contigs. But they artificially dividing the two contigs into two pools. They're referring to HAP1, HAP2, but they're not really representing parental origin. That's why you look at the HAP1 referring to the red color, the HAP2 group referring to the blue color. Then some contigs is actually from paternal, some contig is kind of from maternal, even though they belong to one haplotype group. So they cannot tell the parental origin. But if you have a trio structure, you have parental information, they can do proper so-called truly haplotype resolved chromosome assemblies. In this case, you can see that all the HAP1 is representing, in this case, paternal contigs, and the blue one, the HAP2, really present the paternal the haplotype, the chromosomes. So you can see the phasing using long-range information is still essential, and you wanted to figure out the parental origin, you do need the parental information. So this is a basically evaluation. We evaluate the 40x, 20x, and the 30x. What if you increase the coverage? Would the result be dramatically improved? But actually it's not. You can see that for each of these charts, we evaluate by increased volume of data. The red number representing the data volume we use for current evaluation. Then you can see beyond that, you can see we look at the 1 to 5 parameters in terms of NG50, medium length of the contig. You can see that after 40 using duplex read, you really not increase it for both either haplotype contig or either primary contig. And you look at the use ultra long, yes, you will substantially increase the length, but if you beyond the 20x, beyond the 20x of ultra long, then your performance is slowly saturated already. And for the POC, based on this number, look like we definitely overshoot already. So look like we don't need that much POC data with the 30x, because you can see even with the 20x, you don't see the difference between the 20 and the 30x. You can see, look at the number of sequences. The sequence mean here is a contig, and how much you can reduce the number of contig, because the smaller number is, is good. Then you look at the QVs, the quality of the sequence of assembly. They are basically not changing much afterwards. So here is a summary. What do we got? Yes, we can definitely construct haplotype resolved high-quality T2T human genomes, and using purely single platform. In this is ONT. Yeah, you consist of duplex read, ultra-long reads, and POC data. Both algorithm, high-phasium, vertical, can do a T2T assembly, but definitely high-phasium is much faster than vocal. It'll be probably the good method to do large-scale pan-genome construction. So with this, I'll finish my talk. Thank you for your attention.